Hey guys, so we're here to talk about DNA and RNA structure in this lecture. Hopefully most of this is review for you. So when we're talking about the discovery of DNA structure, we're going to start with the forgotten person, although she's getting a lot more recognition nowadays. In the 1950s, Rosalind Franklin actually performed what's called X-ray crystallography of DNA. And so these pictures that you'll see are actually some pictures that she took. And by looking at these pictures, this is actually one of the key factors that led to the determination that DNA had a double helix structure. Now, if you think about it, she is, of course, not the person that got the Nobel Prize and is given credit for this because her assistant actually took the work and provided it to Watson and Crick, who used her work in addition to the research that they had been doing to make this determination, and they were the ones who actually received the Nobel Prize. So what she found was this pattern that was very regular and repetitive, and it formed this X-like structure, which is what led to the understanding that it was a double helix. So while she was doing her work, another scientist named Edwin Chargaff was analyzing samples of DNA from all sorts of different species, and he made the observation that the percent adenine and the percent thymine always seemed to kind of be pretty much the same, and the same thing with guanine and cytosine. And so he found this rule that's held true for all species that we're aware of at this time. The amount of adenine equals the amount of thymine, and the amount of cytosine equals the amount of guanine. This is the reason that we always talk about how A and T go together and C and G go together, because these are the ones that pair with each other. We should have the same amount since DNA has two strands. Then we have the basic nucleotide structure. And so when we talk about nucleotide structure, we have purines and we have pyrimidines. Purines are going to have a double ring. Adenine and guanine both have double rings. Remember, we're talking about the nitrogenous base part of this. Okay, so they have two rings. Pyrimidines, you'll notice, are all single rings, so they're all going to be one ring: cytosine, uracil, and thymine. Well, obviously, uracil and thymine, it makes sense since one of them's found in RNA, uracil, and one's found in DNA, thymine. And notice the difference between the two is this methyl group here, and that is actually the reason. See, it makes it bigger so it can't leave the nucleus, whereas this one can. So when they pair, they're going to be held together by hydrogen bonds. So you got to think way back when we talked about water and we talked about the hydrogen bonding and that idea where they get that slight positive and slight negative based on the electrons hanging out around more one than they do the other. So those slight charges are what's attracting the pieces and holding the pieces together. But because it's a hydrogen bond, that means that we can also break them apart more easily, which is really critical when it comes to working with DNA. So adenine and thymine are going to have two hydrogen bonds based on where they're bonding, whereas cytosine and guanine have three hydrogen bonds. So we'll do a little quick review. So this review has us thinking ahead for replication, which we'll be doing in the next lesson. But the idea here is, how might the hydrogen bonds actually aid in this process? What benefit would we have by having hydrogen bonds rather than ionic or covalent or something else? And so the idea here is that hydrogen bonds allow for the DNA strands to be easily separated during the replication process. And then we have a DNA sample is analyzed, and you would find that 22% is cytosine. So how much thiamine would you expect in the sample? So remember, cytosine always bonds with guanine. So if we have 22% of cytosine, that means we have 22% of guanine. And out of 100%, we're going to take away the 44%. And then we're going to divide that in half. And so we would expect about 28% thymine. Okay, so moving and continuing with the discovery of the DNA structure, Watson and Crick combined their findings with the findings of Franklin and the findings of Chargaff. So they utilized other scientists' findings and created the first 3D model, which is known as the double helix model of DNA. And so this is what they won the Nobel Prize for. So when we're thinking about the DNA structure and its key features, remember it is a double-stranded helix, so 2 strands, and they forms this curl or twist. The backbone, or these long side pieces, sometimes you think of it as the rungs of a ladder, are a alternating sugar phosphate bond. And the center crossbars are the nucleotide pairings. So that's the A's, T's, C's, G's. They are anti-parallel, so one goes one direction. Notice this one has 3' to 5' going from left to right, and the bottom strand is going from right to left. So they're opposite each other. One strand runs 5 to 3, the other strand runs 3 to 5. 5' is always the free end phosphate group, so phosphate's always pointed towards the 5'. And the 3' end will always have a free hydroxyl group on it, which is on the sugar. So the key functions of DNA, it is, of course, the primary source of heritable information. So that genetic information is going to be stored in the DNA. It will be passed from one generation to the next through the DNA. This is the language of living things. The exception to this is that RNA is the primary source of heritable information in certain or some specific types of viruses, like retroviruses in particular. Instead of having DNA, they will have RNA. So even when we talk about prokaryotic versus eukaryotic cells, remember prokaryotic cells, they don't have a nucleus, they don't have organelles, whereas the eukaryotic do. And so the DNA also has a variation or difference to it. So in eukaryotic cells, the DNA will be found in the nucleus, and it's going to be linear. It's these lines of chromosomes. They form our little X's that we played with when we did the karyotype. Whereas prokaryotic cells, it's in what we call the nucleoid region. So it's kind of like a central location, but there's no membrane, there's nothing to separate it out. And these are typically circular. So we have one large circular, and then sometimes there'll be smaller circular ones that we call plasmids. So prokaryotes, and we'll even get some of those plasmids in eukaryotes as well, where they'll get those little circles of DNA, little small ones. But for the most part, we see them in particular in prokaryotes. As a matter of fact, a lot of our antibiotic resistant genes are found in plasmids that are easily transferred between bacteria. So they're small circular DNA molecules that are separate from the actual main chromosome, whether it be linear ones like we have, or a circular one that we'll find in bacteria. These are separate little circles of DNA. So plasmids will replicate independently from the chromosomal DNA. So we'll go through a replication process with chromosomal DNA, and the plasmids will have their own replication process. Typically, plasmids are found in prokaryotes. That's the most common location. And they have genes that are going to be useful to the prokaryote when it's in a particular environment, but it's not overall required for just general survival. And so that's why I was saying a lot of the antibiotic resistant ones are on the plasmids, so they can turn those genes on just when they're in a situation where they're needed. We can manipulate plasmids in laboratories. We actually do that. And we can remove them. We can insert genes. And we call that a recombinant plasmid DNA when we insert gene into the plasmid. And then we can put it back into the bacteria and have it expressed. Insulin, there's actually processes where they will have vats of bacteria that make human insulin because they have inserted the gene for making insulin into a plasmid, and so when the bacteria are exposed to the right situation, they will make insulin. Bacteria can exchange these genes that are found on plasmids with neighboring bacteria, so they actually will exchange these plasmids. Once the DNA is exchanged, the bacteria then can express the genes it's acquired. So you can't just automatically express genes that you trade with somebody else, but bacteria can do that. So this helps prokaryotes survive in so many different environments. So when we're comparing RNA to DNA, double helix, single helix. CGAT, CGAU. Ribonucleic acid is the sugar. Ribonucleic acid, single-stranded, AUCG. Deoxyribonucleic acid, deoxyribose sugar. Double-stranded, ATCG. So another quick review. What was Chargaff's rule? Chargaff. Oh yeah, wasn't that the dude who was talking about percentages of adenine and thymine and all those? So any species, the amount of A equals T and the amount of C equals G in the DNA. What comprises the backbone of the DNA? That's our sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Which nucleotides are purines? Purines are adenine and guanine. What type of bond holds together nucleotides? That's our friendly little hydrogen bonds. Okay. Summarize the scientific contributions of Chargaff, Franklin, and Watson and Crick. So Chargaff was the A equals T, C equals G. Franklin, she's the one who actually took pictures of that helix shape of DNA. And Watson and Crick created the 3D model of DNA. What are some key differences between RNA and DNA? DNA is double-stranded and adenine bonds to thymine. RNA is single-stranded and adenine bonds to uracil. And then here's a practice-free response question that we should give a try to. Hopefully this helps and you have learned something in this presentation.